let's start up the class today. If you can hear me, you can leave the chat or raise the hand. Give me a signal, then you can hear that. Okay, so it's working. Going to Slack we have. Okay, so few notice for the class. I think this is quite unfortunate things, but the university wants to, lecturers to check up, students' attendance for the final grade. I mean, as I mentioned, that my class doesn't really count the attendance for the score, but if you didn't make up the 1/3 of the class, I mean, there is no way I can give you a grade. This is sort of university policy newly, announced these days. I think it's silly, but I mean, what can I do? I'm also paid off by university, so I have to follow that notion. This is quite sad, but as I instructed last weekend, I mean, I tried to put the notice as possible as I have really good feeling on the day, but I mean, didn't make it. Okay, so I looked up your attendance over the month, and I guess most of the students get the, the attended. So I don't really worry about that, because I mean, if you miss up 1/3 of that, then you're gonna miss the grading. So I think it's okay, and I keep checking that one through the Zoom records, because when I go to Zoom, someone in here, go away. Going to here, previous meeting, and if I go to here, then I can see whether you attend or not, right? So I can download these and merge them and check whether you guys owning the website. Sorry, the class. Okay, so I could see the emails, and I can look up the names. I can check most of the students. I don't know who this person is, so you can rename yours. And the next page is, I mean, it's totally okay. This one is fine. And I missed this guy, so I don't know this person. So if you want to get the attendance check, please rename yours as well. Otherwise, there is no way I can do this for you. Okay. Where am I? Okay, so the exam review. So we have exam last Wednesday. It was good. It was fun. It was great checkup before we totally losing our way. And I think it's quite appropriate, because it's going to be the 10% of your final grade, and I don't think it won't go any harm for your entire grade, because it will be the so part of the sort of the evaluation and also the practice purpose as well. Okay, so this is the distribution of the exam, and this is slightly shifted towards right, because I modified one answer for the one question. So if you get that answer, then you get one more things on that. And then I guess around 25% students get the sort of expected result from the first exam, which is the around 70 to 80% of the total score. And the students in that ballpark, they're not gonna losing their way during the session 3, 4, 5, because the session 2 is sort of the grounding things for the entire semester. So I think if you get the grade greater than more than like 14, I think you're not gonna lose the way a lot during the class. And students between the 8 to 13, you guys have to check up yours before you're going to lose so many things after session 3 and 4. Session 2 is the crucial part of the entire semester. So keep your understanding and leave your questions and discuss whether you understand correctly or not. That's the advice I can give you. And the students, this course between 2 and 8, has to be engaged more than that, and also the class as well, and also the question activities as well. So, if you're not gonna do that, then the distribution going to be much further in the session 3 or 4 exam. Okay, so as I left in the Slack channel, the important things to the exam preparation is the summarize your understanding and also leave your questions onto the Slack channel. And there's no boundary between whatever the language or the format. So you can leave the question and also leave the question in advance than the actual exam period, right? Most of the exam, the situation when you prepare your midterm exam or final exam during the university time, you shouldn't take the I can get the slides or the recorded lecture just before the a week or a few days ago, the exam. And I don't think this is working in my class because every chapter has its own structure, and also all the contents are actually quite tightly related to each other. So if you missed one thing, then you're not going to understand the next thing, right? And some of the students probably feel the urgent just before the exam period, and they think we gather up with friends and put it into the whatever this building, and you guys discuss why that man taught these things at the time, and you guys discuss that probably over the night, but I don't think that is working because the score represent. So the good thing is your instructor, your lecturer is living in the Slack, literally. So you leave your questions just after the class, or at least the week where the class is happening, right? So then you can check up your understanding and just shape your thinking, and that will be sort of helpful for your exam preparation, but also your knowledge as well. So if you go to the questions channel, there is no stupid questions, right? But there are some bad questions, which are the questions just before the exam, right? Those questions not gonna work in the, many situations, right? So times are priceless, and you have to check up your understanding every class. That's important things. So few students are fully engaged over the class, and when I read your questions, I could see that which part of the the understanding, their understanding missing in their learning, right? So like the questions from Gayong or questions from Jiyoung, I can point out that which part you missed in your question or in your sentence, right? And we have around 60 students, but I can, handle that things if you put on your question on this channel, because most of the students, ends up with the same sort of questions in the end. So we can clearly, discuss that point, and also I can, improve my delivery as well, right? So, the students around this area, they well done. They great. These guys missed a few questions, but I think, that could be totally correct if there more time given that. But some, the students, they totally hidden from the question activities. So those things, I have no way I can, induce you to be engaged in this, the activities, right? And also 2 or 3 students gave me an email for the questions. And I always surprised by how students miss the instruction at the first time, and Slack is not really difficult things. You can be on that, and also it's one of the the online platform like an Instagram or Facebook. I mean, if you don't have that account, then I can see that the Slack is difficult for you. But I mean, this is quite easy, right? So you can do like a 3 or 4 clicking by your finger. Okay, so please be on Slack and participate the question activities more actively. That's my all advice for the exam period. And then what is? Okay, I think it's all good. And then going into Blackboard. Where am I? Here, going here. Okay, so exam review. Okay, so the average scores of the exam is 9.6, and the median was 9. So it's about, less than half. And some questions quite tricky, I say, and some questions quite, the straightforward. So, and I can clearly see, which types of the exam, the questions you missed in the exam. So the first format, as I or we practice on the Monday class. Do you guys have questions for that? Or because I see some students raise the hand, or you guys still raise the hand before? After I asking that. Okay, so that's good. Okay, I can go on here. Okay, so where am I? Okay, so let's look at some questions. So this question, this is about the asking gene, and this is fine. And some students missed the part in here. The positions of the genes can be represented by numeric coordinates, right? Represented by numbers. That's one of the high incorrect answers. Okay, and this part. The cytologists who discovered the Mendelian inheritance suggest that the roles of the genes in Mendelian model, right? So the cytologists, they are showing the Mendelian particles using the behaviors of chromosomes, right? And the perspective towards a chromosome move towards genes when Morgan established his experiment on the furflies, right? So that was the sort of the highest incorrectness. And the second question, this is about segregation, and this is fairly okay. And the laws of independent assortment, that is the, I thought this is the mere question for the students, but what students missed a lot was the level of independent assortment can be measured by genomic sequencing data, right? And this is the true because we use the genotype information to whether these two loci or multiple loci, they are linked or not, right? And this is by the chi-square statistics, right? So that was the one part, and the assortment is influenced by the physical distance of on the chromosomes, and that was the question asked whether you understand the recombination happening in which sense, right? So I explained about the shuffling the genomic the blocks on the chromosome, and the probability of getting the same genomic segment or blocks are high when these blocks are tightly closely located in the position, right? So that was one of the highest incorrectness. And the next one is blending inheritance. That was fine. And some students missed this part. Blending inheritance reflects gradualism in evolution. And I think I briefly explained this during the class, but also this is mentioned in the review paper written by Benson, 2001. And this is sort of the concept you can understand from when you compare between the Mendelian inheritance and the blending inheritance. And that is about the how people see the mutation in the history, right? So that was the one thing. And okay, so question 5, and that was one of the difficult one. I think the second difficult one, and what students missed was this. I mean, the original answer sheet, I only put these two, and that one is incorrect answer at the time. And then I reread the question, and I was vague about my the questions and the sentence, because this mutation is a missense mutation that caused the changes of amino acid in the protein sequence, and that means that this will bring the molecular changes in the protein sequence, and this this implicate the dominance in the molecular phenotype. That's why I clicked this as a correct answer. So I mean, I basically give the all correct answer whether you check this or not. So if you get this one and this one, then you get the correct answer, right? And other than that, the multiple choices is quite are quite straightforward. Like this is asking whether you understand the diploid or not. Students quite miss this part in their answer sheet, and that is actually the review one more time in the exam preparation class, right? And question 6 is also quite mere stuff. This is asking whether you understand the probability of the transmission or not, and 50%. This is one of the classic exam I repeatedly put in the exam every year, right? So if you look at these numbers, you are prone to calculate something, but it actually asks you whether you understand Mendelian segregation's probability, right? And this is always equal probability, right? The understanding the equal probability of segregating the Mendelian factors, or in other words, the additive factors, is quite simple, because this is the 50%. But I think this is quite, the in-depth sentence in terms of the polygenic model, because I mean, without knowing that the 50% probability of the additive segregation, I mean, who else suggests the polygenic model in the history, right? So 50%, the same equal probability is, I mean, regardless of the environmental factors, is crucial, right? That's why this is the sort of the basis of the trait the human has in the population. Okay. And the question 7 is the questions about the gender in humans, and the answer is none of the above. And this is the same question from the previous years, right? And also we review these questions in the exam preparation, and then surprisingly, the 10 out of 60 students in this class get the wrong answer. So how on earth these things can happen, but I don't know. Yeah, so students be engaged in the class and be review your stuff before the class, right? I mean, I really surprised by this, right? How you missed this question? Yeah. Okay, so that was the none of above. Good. And this is about the chi-square and the Hardy-Weinberg equilibrium, one of the easiest thing, and also keep asking you whether you understand the equal probability of the segregation, right? Okay, and question 9 was one of the fun thing, and this is asking you two stuff. One is whether you understand the concept of the Hardy-Weinberg equilibrium, and the other concept is whether you understand the dominance is the molecular phenotype, right? So this is basically the whatever the homage of the year's question, right? So I mean, you should get this answer as well. And then also here, and this is that one, and that one is asking you the molecular dominance, right? And this one is asking you the Hardy-Weinberg equilibrium. And then the choices for this sentence is asking you whether you understand the cellular compartments of the genomic DNA, right? So as I explained in the I think probably sex and chromosome, and I explained about the two types of the genomes, right? One is the germline genome, and the other one is mitochondrial genome, right? And germline genome is genomic DNA. Genomic DNA is the the composed of many cells in the tissue, right? So eyes are tissue, right? And within this tissue, we have the multiple cells, right? So that's why we have more many genomic DNA stuffs in multiple cells, right? I mean, if your eye is unicellular, then you could have a few peptide sequence, but I mean, we are a normal creature, right? So we have multiple cells, multicellular organisms like eyes, right? So we have the multiple peptide sequence, right? Okay, so that was the things you missed a lot. And then in this question is it's quite near, and the one is easy to heritability. Is fine, and this is the heritability. You can open up the slide and you can look at the slide. I mean, this is literally open book testing. So I mean, what else you can miss this? And then okay, so that is another interesting question of asking you whether you understand the heritability, whether you understand the phenotypic resemblance using the covariance concept in the familial relationship. And then also asking you about the polygenic nature, right? Okay, so the first answer sheet was asking you whether you understand the segregation, Mendelian segregation, right? 50% chance. And then the next one is whether you understand the heritability concept between the family membership, right? So the relationship between the parent and child kind of compare the dominance relationship, right? So this is the fourth. And the third one is asking you about the covariance of the familial resemblance, right? And that is also asking you the heritability question, so HS. And then Marfan syndrome follows autosomal inheritance. Okay, this is again the Hardy-Weinberg style, the concept question. And that one is also asking you about the polygenic nature, right? So even if you have the same diagnosis, you still have the phenotypic variance observed in the family members, right? Okay, so that was the interesting question. And the question 14 was, this is one of the simple question, right? And question 15 is another, okay, this is something, the average score is 23. Okay, so patients, okay, so this is asking the genotype-phenotype relationship. And this is asking the molecular dominance stuff, and also the same dominant stuff. And this is sort of the same, the examples as SN2A genotype and phenotype relationship, right? And that one is asking you the Hardy-Weinberg assumption, right? And this is asking you the mutation position, right? So we learned about the exon, introns, or UTRs, blah, 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 right? And if you have the sense, or if you look at genome browser closely or by yourself, then you see, oh, this is exon and this is intron. Then that one is in your getting your sense, right? So you have to be familiar with the concept of the exon and intron, and that is also related to your molecular biology understanding as well. Okay, so that was easy. And this is the question 16, and asking you about the features polygenic model. And notoriously, all of the questions are answer. And that is one of the easy thing. And this is the easy stuff as well. And then the question 18, okay, so this is the easiest one. Okay, question 19 was about the Hardy-Weinberg things, right? Okay, I think, yeah, it's the same sort of the evaluation about whether you understand the Mendelian segregation or not, right? So it's fine. Okay, so it's good. And question 20 is a classic pigeon question. This is the annual repeat as well. My Annam Station pigeon question. Geneticists love pigeons. Okay. Okay, so question 20. This is asking you about whether you understand the concept of the normal distribution when we represent the threat value, right? And also whether you can implicate that understanding to the heritability, right? But in this sense, we don't have the generation information, so we cannot implicate that one into the heritability measurement, right? Okay, so that's all good. And as you experience in this exam, but also in the test exam, you cannot get the the good scores. Until you understand very correctly, right? So and also the multiple choices are always a bit of an issue, right? And also the questions are randomly given to your test period. So if you want to, if you want to attempt the sort of plagiarism or whatever the cheating situation with your close friends or your blood, it's not gonna work anyway. So be on your preparation by yourself, and also enjoy the discussion in individual class, and then that will ultimately give you the good exam scores and the test. Okay, so I think that's all, and then let's move on to today's class. Okay, so good. Okay, so we are going to the mutation stuff. Okay, before we go into mutation stuff. So we are moving on to the session 3. So hopefully you really enjoy the session 2 and also get some, the sense of the basic and cores of the core concepts of the human genetics, and you can easily follow up with the session 3. And on the session 3, we are going to learn the concept of the mutation and population, right? So why do I choose mutation? The reason is that the diversity described by Charles Darwin in 100 years ago was the explanation of why the individuals in our population are so varied, and also how we can explain those the varying differences inherited to the next generation, right? And also it's ultimately, consists of the human activities or whatever the traits, or even like human disease. All of these things are we can say sort of the difference, right? And in the sense, one of the key core concept describing and or the basis of the phenomena is the mutation, because we have different composition of the DNA sequence in our genome. And as a biologist, we know that these, the DNA is ultimately put it into the proteins, and these proteins ultimately expressed as a phenotype in an organism, right? So knowing the differences in the mutation, as we can describe the differences in the exist in individuals in our population. But also the question is then how population are so diverse over the time, right? Because as we see in a different population, different individuals, and all of these compositions are quite heterogeneous in some way, right? And if you're, if you're keen on some clinical aspect, you might want to explain the regions of the disease, or if you're interested in the physical activities, and you also want to explain that the differences between the individuals, right? All of these diversities, one part we can explain by the elements in our body, and that is the things we learned in the Mendelian inheritance, like the alleles or genotypes, but more of the modern sense in human genomics, that is the mutation, right? So mutation is a key concept we understanding the differences across the individuals in the population. So I bring that issue into the session 3. Then we look up some the diversities in our population. So in this session, we are learning a few concepts, obviously the mutation and also the mutation classification. And we are learning the linkage disequilibrium in haplotypes. And that is one of the signatures in our genome. And then we are learning the population structure, which is described by the differences in mutation in multiple population, right? And then in the end, we are learning one of the popular examples of the population structure, which is the inbreeding, right? And inbreeding is one of, the notable things in our population stratification. Then again, we also have the review session and Q&A and the exam. And this is sort of the provisionary, the schedule for our session 3 classes. The dates can be changed. Hopefully I can keep up this schedule, but it depends on how we discuss or how we progress over the time. And if the things are all going properly as scheduled, then we're going to have the exam on this day, right? Okay, so once we know about the mutation and population, then we're going to the next session called human genome, how we actually identify the differences in the data set, right? So if you look up the flows of the classes, that will be extremely helpful. Okay, so mutation. So what we're going to learn today. So I'm introducing and also the evolution of the term mutation. As always, the lookup as a historical perspective. And also we are learning the varying definition of the mutation and variant, right? And the third thing is the variant frequency represent a population structure. We are briefly touch on this stuff in the later in today's section. And also we are briefly also discuss about the genomic studies from the recent days and to understand the population structure. Okay, so we still have some the learning materials from the textbook, And also this Genetics in Medicine, 8th edition. This is again very good research, so you can have as a reference. And again, this is always accessed from the our university website. Okay, so let's look at the mutation first. As I spoiled out during the session 2, mutation or the variance is actually the same the meaning. And this is referred to a change of the nucleotide sequence in an organism or in a genome, right? And mutation is an allele in the context of the modern genomics, right? And mutation is also called genetic variant, polymorphism, or genetic variation. So if you look up some, the papers, if you look up some journal articles, or if you look up some textbooks, they are, the mixed use of the definition of the mutation. So some, the journals, they use the genetic variant. Some journals, they use the polymorphism. Some journals, they use the genetic variants, right? But I could say that the most accurate term is actually the genetic variant, right? But, polymorphism is also quite, the famous terminology. Mutation are famous terminology as well. But I mean, the exact meaning is slightly different by themselves. Okay, so let's talk about the change. Change is interpreted in the context of the population, right? Because if you want to say that the change of the nucleotides in an organism, there has to be something we can compare with, right? So if you look at someone's DNA and the someone's DNA has the different composition, then we can call it change, but differences compared to what, right? So change is always has to be, dealt in the, the context of the population, right? So compare your DNA sequence to others one, right? And population can be defined by mutation and have long been studied in the genomics. So I mean, implication of the meaning of the change, which means that the change is the uniquely exist in the in certain population or certain individuals, right? And individuals with the same DNA composition, they are likely to be in the the same population. That is sort of the basic understanding how we see the DNA sequence in a population, right? So population in the context of the genetics, we can define by mutation. And also this is, the long history of the research in our field. We are going to discuss this more further. Okay, so introduction of the term mutation. So when people start using the term called mutation, right? In 19th Europe, there had been debates on how evolution works. So people actually want to know about how the organisms or how our society has been evolved over the time, right? So some people thought that the organisms or whatever the biological creatures, they are suddenly appeared in our population, in our society, and they are seeing this evolution as a radical form. So I mean, something suddenly happening, and there's something is totally different to the population, and they could have advantage or disadvantage in their society. If advantage, there will be survived in the time, right? We call them sortationists, right? Sortationist means that the radical changes of the organisms during the evolution time. In contrast, other people thought that the evolutions can take place slowly, right? So, I mean, we call them gradualists, right? So as I mentioned, that sortationists, they claim the evolution takes place suddenly. So, or we can say sortating, or they describe this evolution as a sporting, right? The same, the words as a sport, right? Sporting, so suddenly happening, right? And so the change is instantaneously transition into the new species, like we see in the movie X-Men, right? In contrast, the gradualists like Darwin, they believe, gradual process of evolution, given a large scale of variability in a population. The gradualists think that, the population is large and they have the wide distribution of the trait value. So if the sortation is working as a sortationist claim, then the trait value has to be discrete rather than the sort of the continuous distribution, right? So always, like, Darwin or the people who follow Darwin's idea, like biometricians, they are on the side of the gradualism as to the evolution process, right? Because they want to describe the continuous perspective of the trait value, right? But anyway, one of the sortationists was the Hugo de Vries. He observed that a new species of the evening prime rose, and that flower was certainly a rosin in his experiment, right? So he basically bred so many, the flowers in his garden. Then one day he saw the variant form. He saw the different flowers, which has the different colors, different shapes, right? And he observed the situation, and how could he, resist the sortationist in the evolution process, right? So he claimed that the evolution takes place suddenly, right? And this is basically the same idea when, the Morgan see the fruit flies experiment and see the red eye flies in his darkroom experiments. Then he described the situation as a sporting situation, right? And I thought that is quite fascinating debates, because the Charles Darwin actually trained his scientific rigor in the people from the gradualism perspective, right? But then when during his the experiment with the fruit fly, he see sort of the examples of the sortation, and then he totally changed it into the new field, right? And that is, I mean, how could we resist that situation if you see that examples by your eyes, right? So Hugo de Vries is one of that person. And Hugo de Vries gave the name Mutation in 1901, and he called it Mutation, and that is seemingly new forms in his experiments, right? Mutate, that is the words of the Greek, and explain these the situation we cannot revert to the previous status, right? And he borrowed that terms and coined a new term called mutation, right? And that means the new form in a population, right? And also, he also brought the term called gene, right? And this gene is, the term tries to explain the Mendelian factors, because the Hugo de Vries also the person only appreciate the Mendelian inheritance, and he tried to explain Mendelian inheritance in the evolution process, right? And the concept of the gene obviously evolved over the time, as we learned in the session 2. And probably around 1930 was the sort of the time people did the hot debates on how genes are working in our evolution, right? And the concept of gene has become more concrete in this area. And I mean, remind you the time is being, the Morgan explained his concept of the gene from the fruit fly experiments. That was around the 1910 and 1920. And afterwards, people, more and more appreciate the Morgan's work, and they are exposed to the situation how evolution takes place by the concept of the gene, right? So genes were regarded as indivisible units of the inheritance. Of course, we learned from the Morgan's experiments, and that is located at the specific points on the specific chromosomes, right? And this is again sort of the transition of the perspective from the chromosome behavior to the level of the gene by the Morgan's experiment, right? And then they are explaining the gene into the 4 criteria. The first one is hereditary transmission and genetic combination and mutation and gene function, right? And all of these things are interplayed over the time, especially in 30s. And after 20 years, central dogma came up, 1950, and they fully engaged in the, the concept of the genetic flow, and people sort of prepare and also give sort of the forerunners of all, molecular biology ideas into the later 20th century, right? And I mean, you probably want to see sort of the floors of the discussions of the Mendelian factors to gene and central dogma and to the molecular biology's appearance, right? So I recommend this review. This is the written by Part1 and Wilkinson in a few years ago. And this is the evolving terms of the gene. And this is quite great review, summarize how people see the gene over the time. So from the classic period of the genetics, then somewhere in the central dogma in here, and then people, and also this is the critical things, 1 gene and 1 enzyme stuff, right? And this is again the important link between the Mendelian factors to central dogma and to the biochemistry. Okay, and this is the central dogma stuff. And yeah. Yeah, it's cool. It ends up with the regulatory stuff at the end. Still, most of the things we vaguely know in our molecular trajectory. So, okay. So if you're interested in biology, then go to this link and enjoy the review. Okay, so good. And then back to the mutation term evolution. So there are some different streams of how people see evolution and the mutation and in the history, right? And one of the view was sourced from the Darwin, right? And that one is the Darwinian view, right? Charles Darwin or people who follow Darwin's view, right? And all of these people, we call it Darwinian, right? And most mutations, in the Darwinian's view, most mutations have an impact on the certain trait, right? So mutation definitely working in the trait value, but for the evolution, the natural selection is the primary force, right? So if the evolution can happen, then natural selection is the actual working horse rather than mutation. So they are more seeing the selection is the critical things. But then post-Mendelian geneticists, which is against the Darwin's view, and they are following the Mendelian inheritance and the mutations sortation view, they thought that natural selection plays little or modest role. So natural selection do almost nothing. But then the occurrence of the mutation would be major evolution force, right? So they think the mutation is doing something important, right? And mutation has to be the key to understand the evolution. And this is the key to make a difference between the individual, right? So there are some theory about that, sortation theory of the Bateson, and the macromutation theory of the Dubris, and mutationism of the Thomas Morgan, right? And we discussed this in the session 2, right? So around that, the Mendelian genetics, the mutation doing something great, and from this notion, people start think about the how the new organism can happen by mutation. And one of the classic example is the hopeful monster proposed by the Richard Goldschmidt on the 1913. And his idea about the macro evolution through the macro mutation. So what that means, if the macro mutation or the mutation something critical if the mutation happen at the critical stage of the development, and that could produce large phenotypic effect, and ultimately that organisms can be remained in the population, right? So if the mutation is happening suddenly in some individual or organisms, and the mutation give a new species or the new traits to an organism, right? So as we see Primrose experiment by Hugo de Vries, if that mutation happen in the flower, then that mutation give you the new trait, give Primrose new trait, right? The same as the Morgan's experiment. So Thomas Morgan sees the red eye fly, right? Red eye fly, the red eye can be happened by some mutation changes, right? And that is again because of the mutation. So in that sense, if you have the mutation in a critical time, critical gene, then that individual, that organism can have the new phenotype, right? So Goldschmidt called these macro mutation are hopeful monsters because they were embodiment occurrence of large phenotypic changes that had the potential to succeed a new species, which is described sortation, right? So change early development does cause loss effects in the adult phenotype because there are chance to have segregation, right? Okay, so I give you one example. So this is the gene called FOXP2. The full name is the fork-head box P2. Why it's fork-head? Because it looks like fork. So this is one of the protein working in the transcription. So we call it transcription factor. So if you learn about the transcription in molecular biology, you probably learn about the RNA polymerase II binding to the promoters and initiate the transcription, blah, blah, blah. And when the transcription occurs, the other transcription factors, which are proteins, bound to the RNA polymerase II, basically helping them, right? So in this situation, the transcription has to be have some, the formation can easily bounce to the DNA sequence. So like FOXP2, it has the shape like a fork. So it can easily bounce to the sequence or recognize the DNA sequence, then it can initiate the transcription, right? So why I met FOXP2? FOXP2 is related to the function to language, right? So during the evolution, there is some point mutation or non-synonymous mutation happen in the species, like the position on 1652. In that position, sorry, not 652, it's I to M changes. It has the differences between the mammoths to primates, right? So they have the unique vocalization given by this mutation. And interestingly, between the chimps and humans, we have 6 different point mutation in FOXP2. And because of these 6 point mutations, we can use language, right? And chimps cannot use language. They have their expression patterns, but they don't have the language, right? They don't have the human language, right? So FOXP2 is okay. Can you hear me? The connection was just gone. I don't know. Hana Science Building is getting really corrupted. Sorry. Oh, well, I mean, this is the TMI. We had an exam last Wednesday, right? And exam was finished at 3 p.m. And from a minute later, the internet in Hana Science Building was totally gone. I don't know why. Someone hacked the internet in our building. I hopefully you're not the person that doing that. But anyway, okay, so back to the connection. So back to the FOXP2. Okay, so because of these few differences in the mutation in the protein sequence, we have totally new traits given in human, right? So this mutation is also arising during the evolution process, and somewhere in the millions years ago, where I don't know exactly, but quite old days, and also these mutations can also driving some differences in the in other protein functions as well. So one of the most well-known examples or the mechanisms of new mutation arising is the gene duplication. So in the ancestry, okay, thank you. Okay, back to your, okay, I think now you can see it. Sorry, my Zoom was totally gone. Okay, good. Now you can see it, right? Okay, so one of the examples of the new mutation is the gene duplication. So in the ancestral organisms, we have the gene in our genome. Then during the evolution process, we have the duplicated genes, which is they happen by gene duplication. So basically it copied the gene into the new place, right? So during the gamete formation, during the meiosis, the gene has been duplicated and it creates the new form. And creating the new form basically gives you the variability in your trait, right? And one of the well-known examples is the synaptic gene. So synaptic gene is the protein working in your brain, working in your cognition, working in your evolution, right? And of course, humans or primates is much higher complexity in terms of the cognition and evolution, emotion. The reason is that we have much more complicated systems in our synaptic genes. We have the thousands of the synaptic genes, and these genes are duplicated over the time. So if you look at the synaptic genes, it actually has the mundane format, which is the cell-cell junction. So synapse is like the cells bind to each other, and that is actually the same format as the cell-cell junction in a unicellular organisms, and that is evolved to have more duplicated forms in their gene or their gene families. And now we have more forms of the synaptic protein complex, like an NMDA or say SK or whatever Markov families you're going to learn in the neuroscience in our department. But anyway, the point is that we have more and more diverse functions because of this the mutation occurrence, right? So in the model of the helpful monsters proposed by Richard Goldschmidt, the new trait or new species can be explained by the macromutation, right? So if you look at these examples, we could say that the Mendelian perspective is correct, right? And this is actually we see from the biology, and we see that this is the correct things, right? But the issue happened after that. Okay, so after that post-Mendelian issue, people also criticize that idea, especially from the neo-Darwinism people. So these papers are including the Fisher, Sherry-Wright, Holden, Dobzhansky, and also the Mutu Kimura as well. And what's the point by these people? So the question was that if you have so many mutations in your genome, and if the mutations are doing something as assumed by Mendelian people, then given the size of the population in human, we're not gonna exist in this generation. The reason is that this is the, where am I? Okay, I didn't put in here, but as the Mendelian's view, post-Mendelian's view, they think the mutations either doing deleterious or advantageous, right? So mutation has to do something, right? But according to Kimura's view, mutations are probably, and most of the mutations are probably neutral, right? Otherwise, I mean, the trait values are too discrete, and also the mutations are too fragile to the environmental situation, right? So given the numbers of the individuals in the human population, and given the rates of the substitution in the human species, we're not gonna have that many deleterious or advantageous mutation in our genome. And the mutation has to be very neutral. The point was that the Kimura thought, you know, the rate of the substitution of the mutation is too high in the biological organisms. So in other words, mutations are frequently happening in human beings, right? So if you have that many mutation in our population, how on earth we could survive in the population, right? So okay, let's put it in the more modern perspective. So we are going to learn about the types of mutation in the next class. But briefly, I can explain to you. We have about 70 mutations per generation. So that means that if you compare your DNA with your mom or dad, you have 70 different DNA differences in your genome, right? So according to the Mendelian perspective, all of these 70 mutations are deleterious or advantageous. You have to be very different from your parents, right? But it wasn't that true, right? We have very similar trait value between the parents and us. The reason is that the 70 mutation is not going to do that many things in our body, right? So only few are doing deleterious or advantageous. Rest, which means that most of the new mutations in the generations, they has to be neutral, right? So that is the, I mean, I wouldn't say this is the most recent view by mutationists, but this is the recent classic view by the mutationist. And the Kimura's view on the neutral mutation has the great implication in our genomics. I explain you in this slide. Okay, so over many decades, biologists have argued about the role of mutation in an evolution or disease state, the clinical studies or other biology studies as well. So understanding mutation and evolution is important in biomedical science. And mutation reflects how much selection is working on traits, and selection represents how many individuals have traits in our population, right? And then it is now clear that all mutation affect on or completely determine our traits, including disease. Okay, so let's put it into more the vanilla, the same. So these days, so 2021, we can do genomic sequencing, right? So if you're going to hospital, you can get the genetic testing for your disease, right? And if you pay more than that, then you can have the genomic sequence of your full sequence, right? So now we're living in sort of these days, right? And people's dream, or at least like you know, the researchers or geneticists or clinicians, doctors, their ambition was to find the mutation can explain disease or traits, right? So in the perspective of Mendelian, if you find the mutation from the person, then you can say, oh, you have the mutation and you have very bad mutation in certain disease, or you have good mutation and that is good for your disease, right? Protecting disease or something, right? So, I mean, surprisingly, most of the Mendelian trained people, they have sort of this binary view, right? But in reality, as we learned the polygenic model, and as we learned that most of the traits are based on the additive genetics, most of the mutations or the DNA sequence changes in our genome are neutral, right? Okay, so let's put the numbers. So how many mutations we have? We have about 3 million mutations in our genome. So we have 3 million differences compared to others, right? If we have the perspective of Mendelian genetics, then we cannot explain these 3 million mutations differences to others, right? Because the 3 million mutations has to have doing something totally different in terms of our trait development, right? But it's actually not. So the mutations on the deleterious or the beneficiary during the evolution process or the trait determinants, there has to be quite few. And there has to be, the additives is working most of the situation in our trait development. So having the concept of how we see the mutations in the trait development or trait variants in our population, you have to understand about the neutrality of the mutation in a population. Otherwise, you are kept by the caught by the ideas of the binary perspective by Mendelian insist. Okay, so that was the one thing. So that's why the definitions for mutation, variant, and polymorphism are varied over the time. The reason is that one of the oldest, the terminology was the mutation. This is the oldest term, and this is initially described the situation for permanent changes in evolution process. Permanent changes is correct, but some mutations are not doing the permanent changes because they are neutral to the population, to the organisms, right? So mutations are not that correct description for that situation, right? So we are describing the DNA changes in our genome by the term called variant, right? And these variants are general description on the changes in the nucleotide sequences. So think about the situation that we have so many DNA changes compared to others, and those are not going to make any differences to others, right? So that's why we describe the situation by the term genetic variant, right? Okay, so, and also the variants are often called genetic variants or variation or genetic variation. These are essentially the same meaning. It's more like a diversity when you describe something. And then polymorphism is also famous and also oldest term. And if you are in medical schools or if you're going to the clinical situation, people often use polymorphism, but it has to be outdated someday. The polymorphism is also one of the old terms that describe the variant with the frequency above 1%, right? But the issue was that the frequency above 1% has the meaning in these days, yes or no. At the time when we describe the frequency in early days, in around 2010, 2011, the frequency of 1%, we care about these numbers, but these days, there is no obvious threshold to describe whether these mutations are common or not, right? So that frequency at the time was to use as the cutoff for the, defining whether the mutation is frequent in population or not, right? Or sometimes people use 5%, but the cutoff is varying over the time. But the point is that when we call variance as polymorphism, when the broadly variance, we know the frequency in certain populations. So if you research the frequency of the variance in certain population, then you call it polymorphism. But I mean, I don't really like the term polymorphism, and this will be, you know, disappear someday. But I mean, people still use it. But yeah, I mean, this is like sort of the transitioning time. Okay, so people are confused. So now we have the guideline for the variant annotation. So if you look, if you go to here, and this is the guideline for the PDF. Okay, great. This is the guideline from American College of Medical Genetics and Genomics. And this is the one of the standard guidelines for the clinical testing. And you probably, you know, going to read someday in your career. And they are concerning about the different terms about, you know, the mutation, variant, or polymorphism, blah, blah, blah. And this is also continuous debate on how we see the mutation in a clinical perspective or, you know, the biological perspective. But I mean, it would be great if you read these things by yourself. And also this is again the transitioning ideas as well. So if you search the ACMG guideline, more recent one that has the more update version, but this issue in 2015, they're clearly discussing about the differences between the mutation, polymorphism, and variance. And one of the classic debates on this issue was, I'm not sure whether I put on here or not. Yeah, this one. This is the one. So this is published in 2014 in Nature. And oh, oops, let's zoom in. Yeah, there was the debates initiate this guideline, right? So there was the initial proposed by Daniel MacArthur and Tish McManoley in 2014. They are proposing the guidelines for the investigating causality of sequence variants in human disease. The reason is that people using all of different, you know, the terms to describe the clinical testing. So people want to make the same and consistent terminology describing the mutation in the clinic. But, you know, I mean, these days people widely appreciate variant is the most broad concept, but still, you know, if I read the documents written in Korean or other lectures, people, you know, often use the mutation and polymorphism. Yeah, so sometimes I argue with that, but yeah, you have to understand that the developing ideas of how we define the variant in the clinical testing. So that's why I put that research in here. Okay, time is almost done. So we are continuing to discuss this section on Wednesday. This is the easy, whatever, the introduction for other topics. Then we are going to move on to session 3 and 2. So mutation classification. So this is the actual, the heavy topics that from the first lecture. And also we are jumping on other, you know, the tricky topics on next week and also the after next week. So like, you know, LDs or haplotypes or population structure inbreeding, all of these things are heavily based on the concept of few, you know, core concepts on session 2. So if you are not confident on the session 2, you can leave your questions on the Slack and discuss, you know, whether you understand the concept correctly or not. Then you can, you know, jump on the session 3. And also the exams on the session 3 is also a mixture of the session 3 and session 2. Of course, the questions is not solely by the session 2. It's you know, the concepts based on the session 2 and session 3 because they are tightly, you know, linked concept. Okay. So I think time's up, and if you have questions, you can leave on the Slack channel as usual. and we're going to continue to discussion Wednesday. Okay, great. Okay, have a great day. Bye.